Hello there everyone, and welcome to this Biology Mind A-Level tutorial on investigating transport across membranes. So just to recap what we covered in the last lesson, we learned about osmosis and how it is the net movement of water molecules through a semi-permeable membrane from a region of high water potential to a region of low water potential. And we also looked at water potential in more detail, in particular solute and pressure potential, and what it means for solutions to be isotonic, hypertonic and hypotonic. So we are going to apply everything we've learned about diffusion and osmosis into investigating how these processes occur across membranes in today's tutorial. So first of all, we'll start by investigating diffusion, and then we'll look at some experiments that investigate osmosis. So let's get cracking. So number one, we're going to have a look at this experiment called the agar jelly experiment. So you might have already done this in your biology lessons, but we will recap it and go through the theory behind it now. So number one, you've got to make your agar jelly. So you make your agar jelly with sodium hydroxide and a few drops of phenolphthalein, which again, you might have met already in biology or in chemistry. Now, phenolphthalein is a pH indicator that turns pink in alkaline solutions. So your agar jelly contains this phenolphthalein and also sodium hydroxide. Remember, phenolphthalein is that pH indicator that turns pink in alkaline solutions. So that's what's in your agar jelly. You then cut your agar jelly into equal sized cubes and place it into dilute hydrochloric acid. Now, the acidic solution is going to slowly diffuse into the agar jelly and neutralise the alkaline solution within the agar jelly. Now, we said that phenolphthalein is our pH indicator that turns pink in alkaline solutions. So that means that the jelly is going to go from pink, when it was alkaline, slowly to a more colourless appearance. Because remember, as the acidic solution diffuses into the agar jelly, it's going to neutralise that alkaline solution. So the jelly is going to become colourless as the phenolphthalein becomes colourless. Because remember, this is phenolphthalein in an alkaline solution, it's nice and pink, whereas it's going to become colourless in non-alkaline environments, i.e. the more acidic you make that agar jelly. So we'll look at that on the next slide. So this liquid here is our dilute acid, our dilute hydrochloric acid. These pink cubes are our agar cubes, which are stained with phenolphthalein, which is phenolphthalein, a bit of a tricky one to spell, but phenolphthalein is going to be pink in our alkaline solution. So our alkaline agar jelly is going to appear pink because it's alkaline. Phenolphthalein is going to stain it pink. This is our dilute hydrochloric acid. We're going to leave these agar jelly cubes to chill a bit in the hydrochloric acid, and what's going to happen is the acidic solution is going to diffuse into the agar cubes and take away that pink colour. The phenolphthalein is going to become colourless. And this is evidence of diffusion occurring. And we know that because our alkaline agar jellies are no longer alkaline. That acidic solution around it has diffused in and neutralised that alkaline solution. Okay, so we can further investigate diffusion by adjusting some variables. So we can adjust our surface area to volume ratio. We can adjust the concentration of either the acid or the alkaline in our solution, and we can adjust the temperature. So let's have a look first of all at varying the independent variable surface area to volume ratio. So if we're going to vary the surface area to volume ratio, what we have to do is we have to cut the agar jelly into different sized cubes, and we can work out each cube's surface area to volume ratio. So for example, a cube, so let me attempt to draw a cube here. a cube with 2 centimetre edges. That wasn't too bad, was it? 2 centimetre edges will have a volume of 2 times 2 times 2 centimetres, which is 8 centimetres cubed. So that's going to be the volume of this cube. So I'll put a little note saying that's volume. And the surface area, which remember is this area of all of the surface around the cube, will be 2 times 2, which is 4 centimetres squared, which is the area of one side of the cube, multiplied by 6, which is 24 centimetres squared. And the reason you multiply that by 6 is because there's 6 faces to the cube. There's 1, 2, 3, and then 1, 2, 3 on the other side. We know that one plane of this cube here, so one square here, is 2 times 2, which is 4 centimetres squared. So the overall surface area of our cube is going to be that one area of that bit of the cube here, the cube surface area here, multiplied by 6 because there are 6 faces to the cube. So that means our surface area of our cube is 24 centimetres squared. So we've got our volume, we've got our surface area, put that in brackets there, which means our surface area to volume ratio is going to be, oops, sorry, my writing sometimes is atrocious. Our surface area to volume ratio is going to be 24 to 8, which we can reduce down. If we divide it by 8, we're going to get 3 to 1. So our surface area to volume ratio of this cube that we've cut here, this cube of agar jelly, is 3 to 1. So I hope that makes sense how you calculate the surface area to volume ratio. So you're going to cut your agar jelly into these different sized cubes. You can work out the surface area to volume ratio just like how we've done here. And then what you're going to do is you're going to place the cubes in the same volume and the same concentration of acid, because those are your controlled variables. They're the variables that you keep the same. And then you measure the time it takes for the different cubes to go colourless. And have a think about it. The cubes with the largest surface area to volume ratio are going to go colourless the quickest. And if you think about that, that's because the cube with the largest surface area, so if I draw a large surface area cube here, so this cube has got a large surface area, this cube has got a smaller surface area to volume ratio, and this cube is going to have that larger surface area for our dilute hydrochloric acid to be able to diffuse in at lots and lots and lots of different points on this cuboidal surface. Whereas this smaller surface area to volume ratio of this cube is going to have fewer points for the dilute hydrochloric acid to diffuse in, which means that the cube with the largest surface area to volume ratio is going to go colourless the quickest, because the hydrochloric acid is going to be able to diffuse into the jelly much, much faster than this cube. So the alkaline jelly is going to be neutralised the fastest by the hydrochloric acid diffusing in. So that's quite neat. So that's how we investigate our varying surface area to volume ratio and how that affects the rate of diffusion. So we're now going to do the same, but we're going to vary the concentration. So this time we're going to cut the agar jelly into equal sized cubes. So again, remember we're trying to control as many variables as we can. So this is one of our variables that we want to control. And then we're going to place the agar jelly cubes into different concentrations of acid. Now, again, have a think about what you think might happen. So the cube in the highest concentration of acid will go colourless the quickest. So that's because we have the steepest concentration gradient, or a greater concentration gradient. And therefore diffusion is going to happen faster between the hydrochloric acid in the solution into the jelly, because we've got a higher concentration of acid, we've got a greater concentration gradient, So the cube placed in the container with the highest concentration will have the greatest concentration of acid being diffused into the jelly. So it's going to neutralise faster. Okay, and last of all, we're going to investigate temperature. So again, we want to control as many variables as possible. So we want to cut our agar jelly into equal sized cubes, so that's our control variable. And we're going to place these agar jelly cubes into containers with the same concentration of hydrochloric acid, so that's another controlled variable. And this time we are investigating temperature, so we're going to vary the temperature. Now you have to be a bit careful here because too high a temperature can melt your agar jelly. So anything above about 65 degrees Celsius is going to melt our agar jelly. We don't want to do that, so don't go above that sort of temperature. But we put our containers in water baths heated to different temperatures, and again we do the same thing. You know it better than me now. We measure the time it takes for the different cubes to go colourless. So higher temperatures will speed up the rate of diffusion. So if I write that here, higher temperature leads to higher rate of diffusion, and that's just because the molecules have more kinetic energy at higher temperatures and they're going to move faster, so our acid is going to diffuse faster into our alkaline agar jelly. So the cube in the hottest container will be neutralised the quickest. So that is how we investigate diffusion. So we're using the agar jellies and we are using an acidic solution, putting our alkaline agar jellies, which have this phenolphthalein pH marker in them, and we're measuring the time it takes for that agar jelly to go from a pink colour, indicating that it's alkaline, to a colourless appearance, which indicates that that alkaline agar jelly has been neutralised by the acid diffusing into the jelly. Okay, so that's how we investigate diffusion. So now let's look at osmosis. So we're going to quickly recap what osmosis is. This might ring some bells from the previous tutorial. So osmosis is the movement of water molecules from an area of high water potential to an area of low water potential. And water potential is determined by the concentration of solutes in the solution on either side of the cell membrane. So we're going to investigate osmosis using plant tissue. So this experiment involves placing plant tissue, so for example we use potato cylinders, in varying concentrations of sucrose solutions, and having a look at the change in mass of those potato cylinders if we leave them in these sucrose solutions for a certain amount of time. So what we're going to do is we're going to prepare a series of dilutions of our sucrose solution, and we're going to place 5 centimetres cubed of each of our sucrose solutions into each of these beakers. So remember, we want to control as many variables as possible. Our independent variable here is our concentration of our sucrose solution, so we're varying that. But we want to control as many variables as possible, so we want to control temperature. We'll want to control the surface area to volume ratio of these potato cylinders. We want to cut these potato cylinders in using the same cork borer. We want to cut them into as equal sized pieces as we can. And another variable that we want to control, which I've written down here, is our volume of our solution. Okay, so we've used our cork borer. We're going to cut out 18 pieces of potato chips, and we're going to dry each of these potato chips as much as possible, again to try and control as many variables as possible and make sure there is a sort of standard water content across each potato chip. So we're going to dry our potato chips gently with a paper towel, and we're going to divide them into groups of 3, as you can see here. And we're going to weigh each group, and we're going to write down how much our set of 3 potato chips weighs across each group. And then we're going to leave our potato chips in the solutions for a minimum of 20 minutes. So cut our potato chip chips, put them in, set the timer for 20 minutes, and we wait. So let's just recap what I've just talked through there, just in writing. So we've prepared our different concentrations of sucrose solutions. You can see them down here. So 0 molar, 0.2 moles, 0.4 moles, 0.6 moles. And then we're going to prepare equal sized pieces of potato chips. So we're going to cut them out using the cork borer. Remember, we want the same surface area to volume ratio because we don't want to alter that here, because here we're having a look at our different concentrations of sucrose solutions. So we keep all the other variables apart from our concentrations constant. And then we're going to weigh the mass of the potato chips, having dried them. And then we're going to put those potato chips in each solution, and we're going to set the timer for 20 minutes. And once 20 minutes is up, we're going to take the potato chips out. We are going to weigh, well, we're going to dry them first of all, gently again, and we're going to weigh the mass of the potato chips again. And then what we need to do is we need to calculate the percentage change in mass. So I'm going to tell you how to do that. I'm just going to write out a little formula for you. So to calculate our percentage change in mass, we take our final mass of our potato chips and we minus the initial mass from that. So that's going to tell us how much that final mass of our potato chip has changed compared to our initial mass of our potato chip before we put it into the solution. We're going to divide that by our initial mass. And we're going to multiply all of that by 100 to make it a percentage change in mass. So if you're asked to calculate this in class, if you're doing this experiment, or in an exam, if this experiment is given to you, if you memorise this formula, you'll be able to calculate the percentage change in mass of our potato chips. And that's going to give you lots of numbers for each of those sucrose solutions, what each of those potato chips, how much they changed in mass. And we can then plot that on a calibration curve, which I'm going to show you on the next slide. So this is our calibration curve. So our calibration curve helps us determine the water potential in the potato sample, because remember, we don't know that. Now what we do is we have, along our x-axis, we have our concentration of sucrose solution, and along our y-axis we've got our change in mass of our potato. So that means that when we plot points on this graph, we're plotting the percentage change in mass against the concentration of sucrose solution. Now let's think about what that means. If we're plotting a percentage change in mass up here, that means that our potato has put on mass because it's a positive value. So the potato has put on mass having been in that solution. Whereas if we're plotting a point down here, we've got a negative change in mass. Our potato has lost mass. So in turn, that also means that the point at which the curve crosses the x-axis is when the sucrose solution is isotonic with the potato samples. So isotonic there is a really key word for you. And all isotonic means it's when the water potential of the sucrose solution is the same as the water potential of the potatoes. So there's no movement of water in or out of the potato. Remember, because osmosis is from high water potential to a low water potential down that water potential gradient. But if we have an isotonic sample, so if our sucrose solution is isotonic with our potato sample, we're not going to get movement of water either way because there's no water potential gradient. Okay, so just to recap, this is where our potato puts on mass, this is where our potato loses mass, and any point on this line is where our sucrose solution is isotonic with our potato sample, and there's no change in mass of the potato. So we plot our points on here. So remember, we had our different concentrations of sucrose in our beakers. We're going to plot how much the potato changed in mass at each of those different concentrations of sucrose. And then we're going to draw on the line of best fit. So I'm going to try and draw a line of best fit on here. A bit tricky on an iPad, but let's try it. There we go. That's not a bad line of best fit. I'm quite happy with that. And what you should see is there's a point at which the line of best fit crosses our 0 point, which, remember, that is the point at which the potato sample is isotonic with our concentration of sucrose. And you should also see that the line of best fit levels off at the last point. So the point at which this line of best fit crosses the x-axis is the concentration of sucrose inside the potato cylinder. So that's really neat, that's really smart. And so you can see here it's probably about 0.28 moles per decimetre cubed, because we can see that it's this point here where our line of best fit crosses our x-axis. Okay, so that's our calibration curve explained. Now, let's just add some labels on this graph to make it a little bit easier for you, and it could be quite handy to jot this down in your notes. So remember, this is the point at which our potato chip is isotonic with our sucrose solution. And we know that potato chips in lower concentrations of our sucrose solution will increase in mass, while those in the higher concentrations of sucrose solution will decrease in mass. So let's think about why that happens. So if we have a dilute sucrose solution, our dilute sucrose solution is going to have a higher water potential than the potato. So if our dilute sucrose solution has a higher water potential than the potato, which way is water going to move? Think about it. So in osmosis, our water is moving from a high water potential to a lower water potential down that water potential gradient. So water passively moves via osmosis to the area of lower water potential. It's going to move into the potato. And this causes the potato to increase in mass. So that's why you see here at this part of the line of best fit, so this part of the graph, where we've got our lower concentrations of sucrose, or our more dilute solutions of sucrose, we're going to have a percentage increase in mass of our potato, because water is moving into our potato down that water potential gradient from outside the potato in that dilute sucrose solution into the potato, down its water potential gradient. And then the same is true vice versa. So in concentrated glucose solutions, which is this part of the graph here, we have water moving out of the potato, because remember, water is going to move from higher water potential gradient in the potato to that lower water potential outside the potato in our sucrose solution. So the potato will decrease in mass. So I hope that makes sense to you. That's our calibration curve explained. If that didn't make sense, just rewind the video and go back and listen to my explanation of what these different points of the calibration curve mean. It's also really important that you understand that this is the point at which the sucrose solution is isotonic with our potato sample. Okay, so that is one way we investigate osmosis. We're now going to look at a second way, and this uses Fisking tubing. So Fisking tubing is an artificial membrane that is selectively permeable because it's got lots of microscopic pores, and this allows smaller molecules such as water and glucose to pass through it. Now, larger molecules can't pass through our membrane, and our larger molecules will be things like starch or sucrose, and they're unable to cross the membrane, whereas our smaller molecules like water and glucose can pass through it. So we're going to prepare 3 equal-sized pieces of Fisking tubing. Again, really important to always mention in exams or in the classroom that you want to control as many variables as you can. So we need our equal-sized pieces of Fisking tubing. We want to place a rubber band at the open end of the Fisking tubing, and this is going to seal that open end of the Fisking tubing. And we're going to fix that in place using a rubber band. And we're going to prepare sucrose solutions with concentrations of 0.5 molar and 1 molar. Now, you may wish to add a food dye to the solution so that it's easier to see within the Fisking tubing. And we're going to pipette in our sucrose solution. So if we start off with our 0.5 molar, we're going to pipette that into our tubing that we've prepared. And we want to make sure it's filled completely to the brim with no air bubbles. And then we insert capillary tubes into each of the tubings. And what we do is we then mark on the capillary tube the level at which the sucrose solution has risen. So I'm going to show you that on a diagram on the next slide. But if for now you just understand that we have this capillary tube in our viscing tubing, and at the end of the experiment we're going to mark the level at which the sucrose solution has risen to. Now, we prepare 3 beakers, each containing distilled water. Sorry, back one slide. So we prepare our 3 beakers which each contain distilled water, 0.5 molar of sucrose solution, and 1 molar of sucrose solution. And we place each viscing tubing into each of the beakers. So we place one viscing tubing into here, 1 into here, and 1 into this beaker. And we leave them for the same amount of time. Again, it's really important that we control that variable. We want to leave them all for the same amount of time. And then we mark the new liquid level on the capillary tube before removing the viscing tubing from its beaker. And we measure that change in the liquid level. So let's just skip onto that next slide with our diagram so that I can explain this a bit more easily with a diagram to help show what's happening. So this is our beaker containing different concentrations of solution. So remember we said that was 1 either distilled water, 2 a sucrose solution of 0.5 molar concentration, and the third option was a 1 molar concentration of sucrose. So that's our liquid in our beaker. We've got our capillary tube here, and what we're doing is we are placing our viscing tubing into our beaker of our solution, and we are measuring where our level on our capillary tube moves up to or down to. So the level, let's think about this, what happens with each of our different types of solution. So the liquid level of the viscing tubing placed in distilled water will have risen because the sucrose solution in our viscing tubing is hypertonic compared to the water. So the sucrose in the tubing is more concentrated compared to the distilled water out here. So we know that water moves from a higher water potential to a lower water potential. So that means water is going to move passively by osmosis from the distilled water in the beaker through our semi-permeable viscing tubing membrane into our tubing where we've got our sucrose solution, which is at a lower water potential. And therefore that's going to cause the level in our capillary tube to rise. So let's say it rises to here, that's going to be the increase in solution moving into our viscing tubing. So with our distilled water in our beaker, there's going to be net movement of water into the viscing tubing via osmosis. Now let's think about what happens if I put not distilled water in my beaker, but 0.5 molar concentration of sucrose. And now we know in our viscing tubing we also have 0.5 molar sucrose. So these two solutions are isotonic to each other. So they both have the same concentration. So there's not going to be movement of water by osmosis because there's no water potential gradient. How smart is that? Now let's vary it again and have a look at what happens if we have 1 molar of sucrose in our beaker. Well, we've still got 0.5 molar of sucrose in our viscing tubing. So that means that this solution is more concentrated than this solution inside the viscing tubing. i.e., that means that our viscing tubing contains more dilute sucrose solution compared to what's outside the viscing tubing. So if our tubing contains the more dilute solution, it contains the solution with the higher water potential, which means that water is going to move passively down its water potential gradient from inside the viscing tubing to outside. And that's because the tubing is hypotonic let's write that word down, hypotonic to the solution outside. See, I spelled that wrong. Hypotonic. So the liquid in the tubing is hypotonic. It's got a higher water potential compared to the solution outside the tubing. So our water is going to move from inside the tubing to outside the tubing, and our liquid level on our capillary tube is going to drop. So that was a lot that we covered there. So remember, we had our solution inside our viscous tubing is staying the same. It's always going to be our 0.5 molar of sucrose. Our solution outside our beaker is going to change. It's going to be either distilled water or 0.5 molar sucrose solution or 1 molar sucrose solution. So those are the 3 varying solutions that we had outside our viscous tubing. And essentially what we're doing is we're looking at how that influences osmosis and the movement of water in and out of the tubing. If our capillary tube level increases, that means we've had water move in from an area of higher water potential outside the tubing into an area of lower water potential inside the tubing. There's a net movement of water into the viscous tubing. Remember that really key word, net, because at the end of the day, water molecules are moving in both directions, but most of them are moving into the viscous tubing, causing that capillary tube level to rise. If we have an isotonic solution between the inside of the viscous tubing and the outside of the viscous tubing, we're not going to have any movement of water in or out of the tubing. And then if we have our 1 molar of sucrose outside the tubing, it means that the solution inside the tubing is hypotonic or has a higher water potential compared to the outside. So again, we're going to get that movement of osmosis, but this time it's happening from inside the tubing to outside the tubing, down its water potential gradient, that net movement of water molecules. And what's that going to do? That's going to cause our level in our viscous tubing to decrease, or our capillary tube level to fall. Okay, so I hope that has explained how we investigate diffusion and osmosis well, and please do go back and watch that, because it is a little bit confusing, but it's really important to get your head around which way those molecules are moving. Remember, with diffusion and osmosis, it's always from a higher concentration gradient or a higher water potential to a lower concentration gradient or lower water potential, down that concentration or potential gradient. And remember, we always talk about it as the net movement of molecules. So remember all of those key words there. And I'd say my top tip is to always have a think about which solution contains the higher or the lower concentration, and then you're always going to be able to work out where the net movement of particles is going. Okay, so well done for today's session, and I'll see you for the next tutorial.